Testing is a very important factor in the COVID-19 response strategy. We need to identify as many patients as possible, as early as possible to prevent further spread. For this strategy to work, fast and accurate diagnostic testing is necessary. Procedures and methods of testing are varied depending on the region, equipment, and the laboratory environment. You may refer to this video, but for actual testing, please follow the proper guidelines in accordance to your products and environment. Collect samples from the diagnostic test. Wear a KF94 mask or personal protective equipment with a higher level of protection. Please wear disposable waterproof long-sleeve gowns or full-length protective clothing, disposable gloves, goggles, or a face shield. When collecting samples, stand sideways to prevent droplets from spreading. In principle, samples are collected by the upper respiratory tract. But if the examinee is persistently coughing or producing phlegm, samples may be collected from the lower respiratory tract. First, get swabs and sample collection containers. Separate specimens should be collected by a nasopharyngeal swab and an oropharyngeal swab, but they must be placed in the same medium. Let's take a look at how samples are collected from a nasopharyngeal swab. The swab should be placed along the human nose to reach the posterior wall of the nasopharynx. Follow the lower part of the nose to reach the posterior wall. When the wall is reached, rotate the swab about 5 times to collect enough samples. An oropharyngeal swab collects sample by pressing down the tongue and swabbing the back of the throat. If the swab is not inserted horizontally or does not reach the back of nasal passage, the test results may be inaccurate. Make sure to hold the swab horizontally until it touches the posterior wall of the nasopharynx. It must be fully inserted and rotated for proper sampling. Drive-Thru screening station was designed to minimize face-to-face contact and prevent infection between health workers and examinees, while reducing the sample collection time to below 10 minutes. The examinees drive their cars through 4 booths. Step 1. Registration. The examinee selects air recirculation mode from the vehicle's air conditioning system and answers the questionnaire. Step 2. Questionnaire. The examiner decides whether to carry out the test or not, and if so, from where to take samples from. Step 3. Sample collection. Samples are collected from the examinees while they stay in the vehicle. Step 4. Vehicle disinfection and guidance. Upon completion of the sample collection, The examinee should disinfect the inside and outside of the vehicle. Examinees are instructed to self-isolate and refrain from having others use the vehicle until the results are out. Walk-through screening stations are used in situations where examinees don't own a vehicle, or when drive-through screenings are not available. Innovative screening stations, such as open-air walk-through screening stations, could be utilized to respond to unique circumstances, including the arrival of large number of travelers to Korea. Insert the swab with the specimen into the viral transport media and carefully break the tip against the cap. Be careful not to contaminate the sample by touching the swab. Close the lid tightly. After sterilizing the sample container with a 70% ethanol solution, mark the patient's personal information and the date of collection on the tube. Then fill out the virus test requisition form. Wrap the sterilized primary container with absorbent materials such as paper towels and place it in a secondary container. Make sure the secondary container lid is tightly sealed and place it in a third container. Write the sender, recipient, and their contact information on the third container. Place the third container in a cooler box and put refrigerant blocks in all four sides of the cooler. On the outside of the cooler box, the label infectious substance must be posted. The recipient, sender, and their contact information must be written down on the cooler box as well. Samples must be transported immediately after extraction and be kept at a temperature of 4 degrees Celsius during the transport. Tests are conducted by research institutes of public health and environment, national quarantine stations, private clinical laboratories and hospitals, etc. If additional verification is required, the Korea Disease Control and Prevention Agency may conduct an additional testing. In the next video, we will get to know about systematic epidemiological investigation.